So first up is Patrick Moldowan with climate-associated decline of body condition in a fossorial salamander.
Thank you, Damien, and thank you, everybody. This project that I'm sharing here today has been a labor of love over the past 12 years or so, with plenty of frozen toes and fingertips to go along with it. Evidence of climate change is unequivocal, and there's growing evidence, especially among terrestrial organisms, of altitudinal or latitudinal distribution shifts. Unfortunately, among amphibians and reptiles and smaller bodied vertebrates for that matter, dispersal is very much a limitation, and so it's a matter of having to cope. With respect to amphibians, we generally expect that there will be range contractions, especially in the north, or at least modifications to their biology along northern range edges or areas where they are already experiencing a bit of a ecological extreme. Especially among anurans, this has been well studied, and we're seeing shifts in phenology, shifts in body sizes between the sexes, and alterations in body condition and phenology. But for a lot of amphibians, which are fossorial or otherwise go unseen, this hasn't been nearly as widely documented. A lot of amphibians and small-bodied organisms, for that matter, occupy microhabitats, and these microhabitats provide microclimates. You know, as relatively large-bodied endotherms, we're fairly ignorant of this, but the thermal buffering that accompanies these microhabitats are really crucial for organisms that exist in environments that are otherwise very physiologically taxing and expose them to ambient environmental extremes. So these sorts of microhabitats can include everything from, epiphytic plants and bromeliads to leaf litters. But here today, I'm specifically interested in animals that occupy burrows that are either directly fossorial, that is, they're burrowing, or they occupy the underground environment. Indeed, about 11% of amphibian species associate with the underground environment, which doesn't sound like very much, but that's 7% of all anurans, over 723 species, twice the number of turtle species globally. For that matter, 23% of caudates and 97% of caecilians are fossorial. So despite occupying this underground niche, these organisms are largely out of sight and unfortunately out of mind. I'm introducing today the spotted salamander, which has a broad distribution across eastern North America. In Algonquin Provincial Park, we're approaching their climatic northern range limit, designated with this small yellow star, and these organisms are largely fossorial, aside from about a 3 to 4 week window in the early spring when they spend their life at the surface undergoing breeding migrations. They spend most of their life underground and presumably buffered against a lot of what the environment has to throw at them. But it's exactly that we are interested in investigating with our longish term data set. So we have a data set that's on reproductive biology of the spotted salamander that spans from 1986 non-continuously until present, with a more concerted effort of mark recapture study that's been going on since 2008. And this work is based at the Algonquin Wildlife Research Station in South Central Ontario in Algonquin Provincial Park. The main study objectives here were to summarize our long-term data set of body condition, but also estimate the relationship between annual climate and body condition, and ultimately use the data that we have right now to predict trends of salamander breeding body condition into the future under realistic near-term climate scenarios. So for each individual that's marked in our study population, we gather a host of morphological measurements, the most important of which here is a body length and a mass metric for each individual. And we use that to measure body condition. Here, for the sake of this presentation, we're considering body condition to be a metric of general health or vigor. And we're using a measure of body condition called Scaled Mass Index, which was published by Pegg and Green in 2009. Subsequent authors have verified that SMI accurately reflects amphibian energy stores by looking at fat deposits in amphibians of varying body condition. But it is crucial to also take into consideration reproductive status, such as gravidity. So here's an example of the same female salamander caught 1 year apart on the right, and you can see she demonstrates vastly different body condition based on whether she is gravid or not. Okay, so to investigate and estimate the role of environmental variables on body condition, we used an information theoretic approach involving model selection as well as model averaging. We essentially formed a bunch of ecologically plausible models using environmental variables and how those might relate to body condition. So we consider temperature, precipitation, and ice-off date, i.e. the start of breeding, as well as winter duration across different seasonal intervals, which may be relevant for these animals, ultimately, and how those environmental variables relate to our response, which in this case is body condition. To provide a summary of our data to date, over the past 12 years of monitoring, we have seen a significant decline in body condition of both males and females. This is taking into account reproductive status for females, so gravidity. And so even while taking that into account, we have seen that both sexes are demonstrating a significant decline. When we ran our model selection endeavor, we found among our set of 17 ecologically plausible models for each sex that female body condition was best predicted by summer temperature, summer precipitation, and winter duration. And that was also true for males. Particularly with the male data set, there was considerable model selection uncertainty. But nonetheless, for males and females, our top ranked model was typically anywhere from 3 to 8 times more likely than the next best competing models. I'd be happy to elaborate on our next competing models, but for the sake of this talk and our time period, I'll move on. When we took our top ranked models and conducted a model averaging to estimate parameters on how each environmental predictor related to body condition in females here in black and males here in gray, we found that mean autumn temperature and mean summer temperatures had strong negative effects on breeding body condition for these salamanders. So that is to say, when mean temperature in summer and autumn are warmer, the salamanders demonstrate a carryover effect in the following breeding season by demonstrating reduced body condition. Likewise, during longer winter periods, salamanders had reduced body condition the following spring. Of course, they're fasting longer, and so they're losing body weight over the winter. And of course, the later we caught salamanders into the breeding period, which is our ordinal date, the less weight they had, presumably because they've been expending energy during the reproductive period. So over the last century, the mean annual air temperature in Algonquin Park has increased by about 1.1 degrees, which is faster than the international average of 0.8 degrees. However, most of the temperature increase that we've seen are actually increases in minimum temperatures, especially in the summer and in the autumn. If you recall from the last slide, it seems to be summer and autumn that is driving a lot of what we're seeing with regards to body condition. So projecting forward into the future, we used estimates for South Central Ontario and Algonquin Provincial Park of climate as it is projected to be in the years 2050 through 2100. This includes, under a more modest scenario, about another 1.5 to 3 degrees increase in temperature and a 2 to 20% increase in precipitation. To put this into perspective, this precipitation variation is within what we typically expect within a year, but 1 to 3 degrees Celsius increase will be above 2 standard deviations increase in temperature for this location. And this is what we found. This heat map here essentially plots what we expect salamander body condition to look like under our y-axis of increasing precipitation, that 2 to 20%, or increasing temperature across a 1 to 3 degree range. And what we see is that if temperature increases an additional 1 to 3 degrees, we expect salamander body condition to decline at a rate of about 9 to 27% declines. In other words, that amount of precipitation increase likely will not offset the body condition declines that we expect with increasing temperature. I perceive these changes in body condition to be non-trivial. These salamanders losing approximately 10 to 1/3 of their body condition is rather substantial. When we looked at temperature profiles of the microhabitat, this involved putting temperature data loggers underground within the fossorial environment that these animals occupy. What we can see are these temperature profiles at the surface in blue. So this would just be at soil surface. Mid depths would be at 0.5 meter, and deep would be 1 meter depths. These are all ecologically relevant values for the salamanders. And you can see how high temperatures get at the surface, assuming these animals are spending time at the surface. When we take these temperatures and convert them to well, these are the average temperatures across the seasons at these depths. When we take those temperatures and convert them to a metabolic rate based on historical literature about spotted salamander rates, we see that assuming animals spend more time at the surface, their metabolic rate is about 1.6 that times deep depths underground, or 1.3 times that at mid depths underground. So there are substantial costs to these animals if they spend a lot of time at the surface, at least energetically and metabolically. So to summarize our findings, we found that body condition has declined over the course of our study period, and this has co-occurred with slight increases in summer temperature. Based on our parameter estimates, summer temperature and autumn temperature seem to be driving this relationship. We're predicting non-trivial declines in body condition into the future with increased warming, and that's unlikely to be offset by these slight changes in precipitation. And our leading hypothesis is that this elevated metabolism or is one of elevated metabolism, and that might be driving the association between temperature and body condition. Of course, there could be a number of other things, such as competition for food, the availability of food, etc. But at least as we are advancing our study forward, temperature seems to be at least a major environmental driver of the patterns of body condition that we're seeing. So what really remains to be seen is we've made a connection between body condition and climatic warming, but whether body condition translates to differences in survival, mortality, fecundity, etc., is really crucial to establish that link, to know whether these individual based patterns will translate to population level patterns. So our ultimate conclusions here are that subterranean microhabitats and microhabitats in general are buffered against the thermal environment, and it's those animals that are surface dwelling that are likely to bear the brunt of climate change in the future. But these organisms that occupy microhabitats, including underground burrows, are not fully insulated from the impacts of climate change. Also, given that there are a huge number of species that occupy subterranean microhabitats, and that these organisms and their microhabitats have been largely ignored, we can kind of consider this a call more broadly to think about life underground and the areas that they occupy. So with that, I'm all done, and I'm happy to take questions if there are any time. Thank you.
Perfect. Well done, Patrick. Yeah, we still have time for a couple of questions. I see Paulson has a question. Would you like to ask it?
Hi. Hey, Patrick. So you said you took gravidity into account, which makes total sense. My question is there some meaningful information in that gravidity term? Like, are you actually seeing differences in the effect on body condition in gravid versus not gravid individuals? I'll leave my question there.
Yeah, no, that's great, Paul. Thanks for that question. You're right. It is really hard to disentangle this, principally because we treated gravidity in this case as a categorical variable, either a female was or was not. I suspect there are females that we catch sometimes that I often call partial post-egg lay, that they have, say, emptied one ovary or oviduct, but not the other. And so there's a bit of a gray line right there. And you raise a great point that those, inherent in gravidity is the fact that females might have varying degrees of body condition, that a female can still be gravid, but just immense resource inputs are happening into reproduction, and her own body condition is compromised or lower because of it. Yeah, I don't know if there's a great workaround with that wouldn't involve, I guess, sacrificing a lot of animals to in some way inspect how much of their body condition is accounted for by having eggs, or or not having eggs, and to what degree having eggs say inflates any measure of body condition. I would love to have a lot of adult individuals that we could capture outside the breeding season, But aside from a handful of adults we get roaming the woods in the late autumn when they come back above ground, we just don't have the sample to look deeper into that. But yeah, a great point that it'd be nice to disentangle those things much better.
So our next speaker is Teagan Netten, and their talk is on habitat partitioning based on local climatic variation does not explain within population variation in morph frequencies of a well-known polymorphic salamander, PLETHODon cinereus.
Okay, great. So yes, my name's Teagan. I'm from the University of Toronto in the Rollinson lab, and this is a project I did as an undergraduate thesis project on habitat partitioning in PLETHODon cinereus. So let's start with PLETHODon cinereus, red-backed salamander. They are a model organism for the study of polymorphism because they are so widespread and they have extremely large population sizes. So it's thought they're likely probably the most numerous vertebrate in Eastern North America, which is kind of, it might surprise some people. You'd think people would have noticed. I'm sure all of you know, but the average person in my experience has no idea that there are salamanders all around them in the forest. But of course, these salamanders in particular are really small, and they spend most of their time either underground or under cover objects, as well as being nocturnal. But because there are so many of them, they contribute disproportionately to their ecosystems. So they're a really important forest floor predator, as well as often being an indicator of ecosystem health. And of course, they're polymorphic. So like many other PLETHODon species, they have this color polymorphism with two common morphotypes, striped, which is this classic red-backed on the top here, and unstriped, which is the black one down below. And these two morphotypes are both common, they're heritable, and they consist of correlated trait complexes. So that means that not only do they differ in appearance, but they also differ along a number of other axes, including physiology, diet, different behaviors, and some other things as well. And so these differences between the morphs might allow them to occupy slightly different niches and therefore be able to coexist. And so the idea is that by partitioning niches, that allows both of the morphs to be maintained within the species. And so, as I mentioned, one of these differences between the morphs is in physiology. And this is a really important factor for this species because, of course, they're ectothermic, but they're also lungless and fully terrestrial. So they only do cutaneous respiration. And because of that, they really rely on microhabitats, cool, moist microhabitats. And when conditions are bad, they will actually retreat underground to find better microhabitats. And so the differences in physiology between these two morphotypes are sort of inconsistent, but generally it seems that the unstriped morph, so the dark one, does better in warmer but wetter conditions. And so in order to look at how temperature and precipitation or moisture affect these morphs differently, one way that that can be done is to look at variation in morph frequency across space and or time. And so as you can see on this lovely map here, the populations of red-backed salamanders vary immensely in morph frequency from monomorphic unstriped, which are the white populations here, to monomorphic striped, which are the fully black circles on this map. And so a lot of spatial correlation studies have found that although the relationship isn't super obvious when you just look at the map, there do appear to be more of the unstriped morph in warmer areas. So generally at lower latitudes and lower altitudes, you get more of the unstriped morph. But in these really broad scale correlations, there's a good chance that there are many other factors that differ among these populations that might be confounding the effects of temperature and precipitation. And so another way to look at this is to see how morph frequency changes through time. And mostly that's been done to look at the effect of global climate change, looking at how morph frequencies have changed over long time periods. And it seems that they haven't changed very much. But there are a couple of populations where it has been reported that the unstriped morph retreats underground earlier in the fall within a given season. So it seems that as temperatures start to cool down in the fall, the unstriped morph retreats underground earlier than the striped morph, again indicating this preference for warmer temperatures. And so my goal with this project was to look within populations throughout the season to see how morph frequency varies with temperature and precipitation in order to see whether those factors differentially affect the two morphs. And that hopefully will give us an idea as to whether these morphs are using different microhabitats and whether partitioning of these habitats is allowing the morphs to coexist within populations. So to do this, I used the Ontario Ministry of Natural Resources and Forestry's Wildlife Assessment Program sites. They have a number of permanent sampling sites in the Lake Nipissing region. Each site has 70 modified cover boards, and every site is visited up to 5 times per season. And so I had a total of 371 visits across 66 sites over 2 years in my data set. So I used the data from 2001 and 2002. And for each visit to each site, the number of red-backed salamanders was recorded, as well as whether they were striped or unstriped. So from that, I could calculate the proportion of unstriped salamanders at each visit. Then I also collected weather station data, from which I calculated the mean temperature and the total precipitation in the days leading up to each visit, as well as getting long-term temperature for each site in order to do an across-site analysis as well. So before I went into my main analyses, I wanted to make sure that the salamanders were using these modified cover boards the way that we thought they were. So we were hoping that the number of salamanders under these cover boards were indicative of salamander activity, so that there would be more salamanders when conditions are good. But it was also possible that they might be using these cover boards as a refuge from bad conditions, in which case we get the opposite signal from what we expected to see. So to just test this, we looked at how the total number of salamanders observed under the cover boards varied with temperature and precipitation. And we expected to see more salamanders when it's wet, so following precipitation. So to do this, we built a number of ecologically reasonable models and did model selection. And we had one model that we decided was sort of our best model that best represented the variation. And that's mod N4 here, which looks something like this. So when temperatures are cold, probably too cold for salamanders to be at the surface, there's not really any relationship. But when it's warm, you see more salamanders under the cover boards following rain. So we took this to mean that we could interpret our results the way that we expected to. So that was great. The number of salamanders that were being observed is representative, or does seem to be representative, of salamander activity. So then we went into our main analysis, the within site analysis. And again, we did model selection. We built 6 biologically reasonable models looking at how the proportion of unstriped salamanders this time varied with temperature and precipitation. And here we expected to see more unstriped salamanders when conditions were hot and wet. But this time, when we did model selection, we didn't really have any good model. So as you can see from the AICC weight here, it's pretty evenly distributed across all 6 models. And so there didn't seem to be any of these models that really adequately represented the variation or explained the variation. And when these the effects of each of these variables was averaged across the models, we found basically no effect of any of these variables. The effect was either 0 or very close to 0. So doesn't seem like any of these variables are having any effect on the proportion of unstriped salamanders. So then to finish up, we wanted to just do an across-site analysis to compare to see if we would get the same results. This time we modeled a mean overall proportion of unstriped salamanders from each site with the long-term average temperature for each site. And again, we expected to see more unstriped salamanders at hotter sites. But although there is a line drawn here, as you can see, there's a huge amount of variation. Even at the same temperature, you can get a huge amount of variation in the proportion of unstriped salamanders. And so there was really no clear relationship here. This was not a significant relationship. So there did not seem to be an effect of, there didn't seem to be more unstriped salamanders at warmer sites. So overall, what we're seeing here is pretty much no effect of temperature or precipitation on whether these morphs retreat underground differentially or are using different microhabitats. There doesn't seem to be a strong effect of temperature or precipitation. It might be a factor in the maintenance of this polymorphism, but it's definitely not the only one. And this was sort of found with both our within and across site analyses. So we've got agreement there. And so it's still sort of a mystery. What is maintaining this polymorphism? And there are very likely other factors at play here. Maybe genetic drift has more of an effect than is presently thought. Maybe there's something else like apostatic selection based on predation that's at play here, or maybe something that no one has even thought of yet. So it's really interesting, and I would love to find out more about it. So I'd just like to acknowledge that this study took place on the traditional lands of the Huron-Wendat and Petun First Nations, the Seneca, and most recently the Mississaugas of the Credit River. Like I said, this project was done as an undergraduate thesis at the University of Toronto. Thanks to all of the people who collected data for the wildlife assessment program, and special thanks to these people right here and all of the Rollinson lab for their help.
I have a question. So you paired your sites with weather station data, right?
Yes.
Because one of the challenges I found with weather station data is like maybe it doesn't account for the microsite variation. Have you considered what the differences would be if you paired them with like a temperature and humidity logger on site?
Yeah, so unfortunately, I couldn't collect data myself for this project because of COVID. And the only on-site data that we had was like a yes or no, a presence or absence of rain in the previous 24 hours. We didn't have on-site temperature or precipitation data. And that's definitely a limitation of this. I think it would be super interesting to look at on-site temperature and moisture values for these visits, because like you said, they use like I said as well, they use microhabitats, right? And so that's definitely probably more applicable than these broader scale temperature and precipitation measures.
Perfect. I think we have time for one more question in the chat from Barbara. And they asked, they said, excellent presentation. I'm a big fan of polymorphisms and trying to figure them out. Do you think color stripe has an adaptive function in terms of avoiding predators? Yeah, so a lot of the time when there is this sort of visual difference, right, this color difference, you'd think that that is an important, like that there's some visual aspect. And it's interesting because they are mostly nocturnal. And so that may not play as much of a role. In terms of predation, there have been mixed results. So I think some studies have found that the unstriped morph seems to be more like camouflages better, can hide better from predators, but other studies have found that there's not really any difference there. And the sort of other idea with this predation is that it's more the common morph is more likely to be predated on because predators have this image of their prey in their mind. And that image tends to be based on whatever morph is more common. And so then that might lead to frequency dependent selection where whichever morph is more common gets preyed on more.
and then becomes less common, and then it sort of cycles. So yeah, maybe, but it's sort of unclear, I guess.
Our next talk is going to be a 5-minute talk, and it's by Jordan McDonald, and the talk is titled Larval Growth Rate and Phenology of Emergence from Natural versus Constructed Ponds in an Endangered Salamander Complex on PLEE Island, Ontario.
All right, so thank you for that introduction. I'm Jordan McDonald. So I am a master's student in the Murray Lab at Trent University under the supervision of Dr. Dennis Murray and Dr. Thomas Hossie. To give you a little bit of overview of my project. So the project is focused around the federally endangered species of Ambystoma salamanders found on PLEE Island in southern Ontario. And as most of you know, the loss of wetland habitats across Ontario is having negative impacts on the amphibian populations which rely on these areas for their continued survival. So to help mitigate these effects, several organizations have begun to construct artificial ponds in order to create new areas for reproduction and larval development. So this is the case on PLEE Island, and which is home, like I said, to many species of federally endangered Ambystoma salamanders. And these species exist as a mixed ploidy complex, which is composed of both sexual and unisexual individuals. So the goal of this project was to determine the conservation value of these constructed ponds to examine if they adequately replicate the conditions needed for the conservation of these species. And shown here on the slide in this photo is one of the ponds that was constructed last year by NCC, and it was one of the focal sites for this study. So we had 6 focal sites, 2 of which were natural ponds, and 4 were constructed. So that was the site shown on the previous slide. It is also shown here in some of the photos. So before we began doing any work with the juvenile salamanders, we had to first determine which ponds contained larvae and then take some measurements from those. So we did regular dip netting, which you can see in one of the photos here, to determine the larval growth rate, and as well as taking genetic samples to then compare to the juvenile stage. So once we began to believe that the larvae were going to undergo metamorphosis, we would begin to erect our pitfall traps and coverboard arrays. So as you can see here, we at 2 of the sites, we were able to fully encircle the pond, and then we would sink paint cans in the ground for our pitfall traps. And those traps are checked every 12 hours as per the MNR regulations. And once we found juveniles in those traps, we would then record the timing of emergence, size at emergence, as well as marking them with a fluorescent marker and taking genetic samples. So unfortunately, we couldn't fully encircle all of our sites. So at those other sites, we had to supplement the areas that we couldn't fully encircle with pitfall traps with coverboard arrays to kind of make sure that we can get as large a sample area as possible around those other sites. So since we just began this project this past summer, we haven't yet had a chance to analyze any of the data or begin the genetic analysis as well. But some preliminary results that I can share with you today is that the pond that I previously mentioned that was only constructed less than a year ago has already been colonized. It contained the largest larva of all of the other focal sites that we studied. And all of these larvae underwent metamorphosis nearly a month prior to the more natural sites, which was very interesting to see. And it's very good news for conservation. It means that this pond was created well, and that it is being used already by the salamanders to hopefully give them a new area for future reproduction. So we also determined that metamorph dispersal from the ponds, it was not skewed towards forest cover, particularly in ponds that are not embedded right in the forest. The metamorphs for juveniles seem to emerge at all sides of the pond, not necessarily just on the sides that were bordered by forest cover. And unfortunately, like I mentioned, the genetic results are not yet complete, so I cannot say much about that aspect of the project. So for the future goals for this project is we wish to use all of our findings to improve artificial pond construction. And we also wish to help improve the pond construction for the benefit of these at-risk salamanders. And we also want to provide new insight into the competitive dynamics among members of this Ambystoma complex. So I would like to thank all of the wonderful organizations and people that were involved to make this project possible. We had field support from Scales Nature Park, as well as other students from Trent University and the Murray Lab. We had logistical support from the NCC, and also would like to thank the Ontario Species at Risk Stewardship Program, and Scales, Trent University, and the Murray Lab. So thank you for listening to my talk, and if anyone has any questions, I'll be happy to answer them.
Next up, we have Evan Barr, and their talk is identifying multiple clonal lineages in isolated unisexual ambystoma communities.
So yeah, I'll be presenting on some of my work for my second chapter of my PhD thesis here today. So normally when I give this talk, I have to, break down the reproductive system for the unisexual ambystoma. Luckily, yesterday, Dr. Greenwald provided us a good synopsis, but here's sort of my own little diagram interpretation. As you can see, it's rather complicated, and this is only when involving 2 of the potential hosts out of the 5 that are available. So last time I presented here, I was able to show how local unisexual communities were structured via the interplay between the complex reproductive system of the unisexuals and the local host availability. However, this only manages to deal with one aspect of diversity, as it forgoes looking into the deeper genetic structure that sort of underlies everything that's going on. Furthermore, this community analysis doesn't help us determine much about potential connectivity or variation within any particular genomotype. Being able to investigate these details would no doubt provide meaningful guidance for conservation efforts. And to this end, I'll be, as mentioned, presenting some preliminary genetic results for my second chapter. Now, to investigate our genetic diversity in this clonal system, we've been using microsatellite markers. As opposed to more traditional markers where alleles are determined by sequence, microsatellite alleles are based on length. Thanks to internal tandem repeat sequences or nucleotide motifs, microsatellites have a far higher mutation rate and tend to also have much higher allele variability. This means that even between clones in the same egg clutch, we can find different alleles at the same locus. And even within the same clonal lineage, we should still expect close genetic similarity compared to individuals of different potential lineages. So first, I think I should develop some basis for making this claim that we even have multiple lineages. So microsatellite markers allow us to compare genotypes through mutation steps, where one mutation step is one motif difference between alleles. Essentially, this amounts to a distance metric, and by doing such pairwise comparisons, a frequency distribution can be attained. As mentioned previously, animals within the same lineage should have very similar genotypes, and thus a low average pairwise difference. Alternatively, when comparing animals of 2 different lineages, genotypes will consistently have higher pairwise differences. So when we see something like this, where there are multiple peaks, this would suggest the presence of multiple clonal lineages in our data set. Now, when looking at our entire diploid sample, though, we see that we have multiple peaks cropping up, indicative potentially of different clonal lineages. So going forward, we'll define a lineage based on a 3 mutation step demarcation, meaning that any genotypes that can be linked by 3 or fewer mutation steps, either directly or through intermediary genotypes, these will be all lumped together as part of the same clonal lineage. Now, just to be thorough, once we've analyzed all of that, we come to find that we had 5 distinct lineages, each of which made up at least 1% of our total sample. Every other individual that either did not have a complete genotype or did not constitute or was not part of a lineage that constituted more than 1%, all got dumped into the 0 category. But using this, using our 5 lineages that we've determined, we can start putting all of our diploids into a PCA. And when we do that, we find that our lineages clump together pretty nicely. And just as a third measure, one more step, we also used Breuveau distance, which is specifically designed for microsatellite data. And once again, our lineages line up really nicely. But this was just to confirm that this perspective was appropriate. Now we can take this and investigate how these lineages fall out across the island. So using our determined lineages and identifying all of our samples at each site, we can sort of represent each site as a pie chart, with each section being a chunk, or each section representing the relative proportion of a particular lineage. So now this still begs the question, like, is there any commonality that is shared between sites with similar lineage representation? So to do that, we can construct this dendrogram based off of a Bray-Curtis analysis as a sort of beta diversity measure between each site. And now using this, we can sort of explore a few different hypotheses. In my previous presentation, in my first chapter work, we found that host availability was a strong indicator of unisexual community structure. If this is the case, we should expect site B3 to be noticeably distinct from all other sites, as it is the only site where A. laterale is the exclusive host. Additionally, site D3.1 would be an intermediary between B3 and all other sites because it is the only site with both potential hosts. However, neither of these predictions really pan out, as both D3 and B3 are more similar to taxonomy exclusive sites than they are to each other. Next, we can maybe look at history. Prior to human development and drainage of the marshlands, PLEE Island was 3 separate isles. If lineage representation were based on this historicity, we would expect populations on the same isle to be more similar to each other than to populations on other isles. Additionally, we might expect the middle isle populations to sort of be intermediary between the north and the south. But again, we don't see this pattern. In fact, the northern isle populations are split across the 2 primary branches of the dendrogram, suggesting high dissimilarity. Adding to that, southern isle populations are found to be more similar to other northern isle populations rather than to middle isle populations. So this historic landscape structure also doesn't seem to quite fit the bill. One last argument that could be made is just it's very localized. And so based off of this, this perspective seems to be the most appropriate, as neighboring sites are shown to have high degrees of similarity. The only pair where this doesn't quite hold is with D6 and D7, though this may be due to a low sample size and low representation of alternative lineages. There's also some concern where regions are in close proximity, namely D3 and D1, are quite dissimilar. That aside, these patterns of localized similarity suggest that lineage analysis can offer insights into potential connectivity. These Alternatively, these patterns could represent differential selective pressures on each lineage, supporting a more frozen niche hypothesis model in unisexual embystoma. Now, next, we can also wonder, where are these lineages coming from? This system we know is technically monophyletic, and yet genetically they're very distinct on this island. So we might assume, again, sort of a historical look, arguing that PLEE Island happens to be a focal point of multiple secondary contacts. Now, if this is the case, we might expect to see similar high lineage diversity at other sites on the mainland or on other sites in Lake Erie. An additional explanation would be from the proposed diplotization mechanism in the unisexual reproductive system. Typically, different lineages in clonal vertebrates are the result of independent hybridization events, but again, ours are monophyletic. It's a single hybridization event. So this hypothesis that diplotized LT offspring could come from symmetrical LLTT tetraploids offers a viable explanation, especially considering the requirement that both hosts need to be present in order to produce these symmetrical tetraploids. Moreover, PLEE Island stands out among unisexual sites for its high abundance of diploid animals, making up almost half of our entire population size. Far more often, unisexual populations are dominated by triploid animals. So this diplotization process provides a source for a diploid population to essentially keep regenerating. With disomic inheritance, randomized chromosome sets for each haplum, and crossover during meiosis, diplotization would produce novel LT genotypes. Furthermore, it would be expected that younger lineages have lower proportional representation, and this pattern can be assessed by testing for a Pareto distribution with individuals highly skewed towards one or very few groups. And this seems to be the case in our data set, as our p-value comes out to 0.003. So this is all still preliminary. We're still looking to include 2 more years worth of samples. So we're looking forward to adding that. But beyond that, we also have some extensions to this. First, we're increasing our number of loci from 7 to 14. This can provide us higher resolution data that may help tease apart some potentially cryptic lineages. For instance, one lineage that we currently have appears to maybe have 2 distinguishing subgroups as seen on this table. Additionally, we'd like to look at some mitochondrial sequencing. While it's unlikely that there's been much differentiation within 4,000 years, if these are the result of a secondary contact or lineage or novel lineage production, we can test these different hypotheses based off of this mitochondrial sequencing. Next, we're also looking to investigate some life stage comparisons, namely adult to larvae. We might end up finding that some lineages just have better survival rates than others, and this may even vary from site to site, suggesting that there's some niche differentiation between these lineages. Lastly, with our Breuveau distance, we can also incorporate our polyploids, and by doing so, we can essentially ratchet up our lineages, not just from the diploid stage, but also to triploid, tetraploid, and maybe even pentaploid levels. So that's what I've got for now. I hope next year we'll be able to produce some more finalized results on this. But for now, I'd like to thank, of course, everyone in the Murray Lab that has helped with the field work, and the Murray Lab in general for their constructive feedback and support. We've gotten significant help from Nature Conservancy, finding sites. Scales Nature Park has helped us out in the field, and we've gotten our funding through the SARSP as well.
Great talk. Thanks, Evan.
Yeah.
We have a question in chat from Karina's, who asked, what was your sampling method?
What was my what? Oh,
sampling method.
sampling method?
Yeah.
Okay. Yeah, so we took tail tissue samples from each of our individuals. Our adults are all VIE marked, so we know if we catch one, we've already sampled it, we don't need to resample it. For our larvae, previous years, we'd only go in and sample, or the first set that we sampled, the first set of 30 individuals that we sampled, we would genotype those and use those genotypes for our larval data set, and everything else was just sort of a sequential time series.
Oh, there we go. We got one from Katie, Dr. Greenwald, and she asked, curious if the loci you're using only amplify in T genome. If not, curious how you'd extend your analysis to unis, since you would expect different allele size in L versus T.
Well, we have some loci that only amplify in laterale, and we have some loci that only amplify in texanum, and we have a set of 2 that amplify in both species. So if we get our laterale exclusive and our texanum exclusive both amplifying, we know at the very least it's a unisexual. And then from there, with our ambiguous loci that can amplify in both species, we can get an estimation of their ploidy level at the very least.
Next up, we have Jana Bergman, and their talk is assessing the effects of prevalence and species distribution model for long-toed salamanders in southwestern Alberta.
Hi, my name is Jana Bergman. I'm really excited to talk to you today about some undergrad work that I did for an independent study on modeling suitable habitat for long-toed salamanders in southwestern Alberta. So the University of Lethbridge is located on traditional Blackfoot Confederacy territory. We honor the Blackfoot peoples and their traditional ways of knowing and caring for this special region of the world. So some background to start off with. As most of you know here, the amphibian populations are experiencing the highest global decline out of all other animal taxa. And the leading cause to this unproportionately high decline are due to a loss of suitable habitat through habitat destruction and modification. Therefore, it's important to be able to model these species distributions, to be able to gain a better understanding of the factors that shape them. And one way to do this is through geospatial approaches. So a specific type of geospatial approach that is increasing in popularity is generating species distribution models. Quickly, the idea here is to use the presence records of where the species has been found to extract information from GIS datasets that for the environmental conditions that underlie the presences on the landscape. And then with this information, we generate a model that differentiates between those conditions where the species is found and those of the background conditions or where the species is absent. It's also important to note that often many assumptions are made when generating these types of models. So testing the settings that are used to generate the models is really important to do. And then after the models are generated, we then project them across space to illustrate places where the environmental conditions are likely to be suitable habitat for the species under study. So when generating species distribution models, a setting that is often overlooked is prevalence. Prevalence can be defined as the frequency of a species occurrence among surveyed sites across the landscape. And throughout published literature, this has had inconsistent results, and that's due to usually when sample sizes are small or when there is biased influences in the sampling techniques. An additional valuable step when generating these models is to use independent data to validate the model predictions. This also isn't often done, as seen in a study that my supervisor was involved in, where they did an extensive Web of Science search for the literature that used independent data to validate model predictions. And they only found 101 studies that did do this. And of these studies, a limited 53% were found to have AUC values that returned greater than 0.7, which indicated that those models were reasonable. So for my study and the models I generated, we focused on the long-toed salamanders. They're, of course, small pond breeding amphibians that are a species of special concern in Alberta. They have a limited and narrow distribution in Canada, and this is especially seen in southwestern Alberta, and it also corresponds to the eastern edge of their known distribution. So my main objective for this study was to model the distribution of suitable habitat for long-toed salamanders in southwestern Alberta. While doing this, I also wanted to test the impact of assumptions about prevalence on modeled predictions, and then evaluate the models with independent data on occurrence records. So the data that I used to generate the models consisted of 4,821 locality records that were gathered from the entire species range, as well as 21 bioclimatic variables that were used for the environmental predictors. I used the algorithm MaxEnt, where 10,000 background records were generated from a 200-kilometer buffer that was formed around all of the input localities. And then as per my first objective, I generated different models with different prevalence settings. MaxEnt has a default prevalence setting of 0.5. So in addition to this value, I tested 0.4, 0.6, and 0.7. These prevalence values were chosen based off of what is known about the long-toed salamander's rarity, local abundance, and the sampling efforts that were used to generate the data sets that I used. So there are many other types of decisions that need to be made when generating these models, and I will further be exploring some of these in my undergrad thesis, but the models that I'm about to show you are just based off of these decisions here. And so as per my second objective, I did use two different independent data sets to validate the model predictions. Dataset 1, which is the blue points on the map here, is composed of sites used in an amphibian monitoring program based in Waterton Lakes National Park. And then the second data set, which are the red points on the map, is composed of survey sites from across southwestern Alberta, and that was from a former Master's of Science thesis. So in contrast to the Waterton Lakes National Park dataset, some of the sites in the second dataset have had ongoing and recent fish stocking with non-native fish populations. And this is an important difference between the two datasets, which I will come back to in just a couple slides. So these are the MaxEnt results that changing prevalence has on the model over the entire region of the long-toed salamander's distribution. The blue and colder colors are areas of lower predicted probability of occurrence, and the warmer colors are areas of higher predicted probability of occurrence. And as you can see, changes in prevalence doesn't have a large impact throughout most of the region. It's relatively consistent. However, in some areas, specifically at the edges of the range, including the area in southwestern Alberta, it does show that prevalence does have a larger impact on the predicted probability of occurrence. So just to zoom in on southwestern Alberta, as you can see in this region, specifically at the edge, prevalence setting does have more of a dramatic impact on the model predictions. As the prevalence setting was increased, the area that had higher predicted probability of occurrence also increased. So then I wanted to formally compare the relative differences in probability of occurrence among sites under the different prevalence models, and I did this using Spearman's rank correlation. So this plot shows the ranking of test sites of prevalence of 0.4 versus prevalence of 0.5 for the first data set, and it is a high correlation between the two prevalence settings. I saw similarly high correlations when other model predictions were compared in this way. And these results suggest that even though the magnitude of probability of occurrence increased with higher prevalence settings, the relative differences among sites were unaffected by prevalence in this region specifically. So with respect to sub-objective 2, independent data can be used to determine if the models are any good. And for a threshold independent evaluation, a metric called area under the curve, or AUC, is commonly used to measure the predictive power of a model. So AUC values of greater than 0.7 are commonly used to consider models as acceptable. And as you can see here, when validated with the independent dataset 1, 3 out of the 4 models did have AUC values that were greater than 0.7. And so in general, these can be considered acceptable. However, they're not the best models. The prevalence setting of 0.4 and 0.7 did have the best performing models. And then the default prevalence setting of 0.5 actually returned an underperforming model. I'm also showing the percent of correctly classified sites, which is a threshold-based metric, and this percentage was high for all of the models, and they all performed equally well with this statistic. So an interesting result, though, is that when we validated the models with the second independent data set, all of the AUC values were returned around 0.5, which is no better than a random model would perform. The percent of correctly classifieds also dropped quite a bit to be around 50% for all of the models. So these results are really different from what we saw with the first independent data set. So why are we seeing these differences? It's most likely due to some of these sites, as I previously pointed out, being recently stocked with the non-native fish populations. This is a human-introduced biotic variable that I didn't consider when generating these climate-based models. However, the effect of this biotic interaction has resulted in sites that were most likely considered environmentally suitable by the models, and in reality, they were unoccupied by the long-toed salamanders. So some previous work from a 2004 Masters of Science thesis by Kim Pearson has shown that when long-toed salamanders are grown in a controlled environment in the presence of fish, the mean survival of the salamanders is significantly reduced. When fish were absent from the environment, the salamanders had just under a 50% survival rate. And then when minnows and trout were introduced, the survival rate dropped to be below 10%. Therefore, this does suggest that the exclusion of salamanders at sites in the second data set, which I pointed out were likely suitable sites and likely once inhabited salamanders, are no longer suitable because the salamanders are now predated on by these non-native fish populations. So to bring it back to the main implications, first, the prevalence setting can affect the overall amount of area where the species is predicted to occur in some regions. And this is specifically seen at the edge of a distribution, although the overall rank order of the predictions for sites does not seem to be impacted by prevalence. And therefore, when using MaxEnt specifically, the default setting of prevalence should not be used. Rather, testing the effects of prevalence in sensitivity analysis should be done. However, when just prioritizing sites, such as in conservation efforts, this doesn't seem to matter as much. Second, I showed that different independent data sets can lead to different conclusions about the performance of the models. Therefore, independent data sets that are used to validate model performances need to be carefully considered, especially in regards to human changes to the biotic environment, such as we saw with the fish stocking, which can cause independent surveys to be misleading when assessing a model's performance. So to bring it back to amphibians in general, this is a map from a CLIMATIC report that shows the current Alberta fish stocking sites as the blue points, and the only other salamander found in Alberta, the western tiger salamander, as the red points. And as we can see, this species range is also being impacted by the immense amount of fish stocking that takes place in Alberta. This is ultimately leading to contractions in these species distributions, which is a conservation threat that should be considered, especially when modeling suitable habitat for species of special concern, such as the long-toed salamanders. So thank you for watching. I acknowledge all of those who have helped with data collection and information and funding, and also the Lehigh Lab for their discussions and support.
Well done. Thanks, Dana. Very interesting stuff. I think we have a question from Gabrielle, who asked, why did you select a buffer of 200 kilometers? What's your reasoning for that? Was it an abstract question, or is there empirical evidence suggesting that there's an appropriate buffer size in salamanders?
So it was kind of chosen just to limit the area around it. Usually, from other studies that I have read, buffer sizes are usually determined by their range size and how far they can travel. And so I think going forward in our, because I'm going to be working on this for my undergrad thesis as well, so it will be something to consider there to get the models better performing for that.
Awesome. And maybe I'll just throw a question in there. Would you say they were stocking non-native fish in Waterton National Park?
They did prior to 1970, and then they stopped, but the other data set does have sites in the Castle Special Management Area, and they do stock those lakes annually with different fish populations.
Of course. I can imagine the landscape level ecological impact of fish stocking.
Yeah, it is. Thank you.
Thank you. So our last talk of this session is going to be a 5-minute talk by Graham Smith. and their talk is entitled Identifying Suitable Areas for Habitat Creation for Ambystoma Salamanders on PLEE Island.
So I'll be presenting some more research done on the Ambystoma complex on PLEE Island. So this is one aspect of my master's research. So this project focused on habitat differences between Ambystoma salamanders on PLEE Island. In general, these salamanders require ponds to reproduce, and they must be fishless for egg and larval survival. These wetlands should also be close to forested terrestrial habitat. So smallmouth salamanders use several types of moist forested habitats and occasionally agricultural land when it's adjacent to suitable breeding ponds. They generally don't travel very far from their breeding ponds. Blue-spotted salamanders are more tolerant of a broader set of environments, including dry, sandy conditions, and they're more willing to travel across open fields. Unisexual ambystoma are assumed to use similar habitat to their sperm donors. Unisexuals with genomes from the smallmouth salamanders have been found to be restricted to areas with clay-based soils, and recent work has indicated that the so-called hybrid nuclear genome of unisexual ambystoma has resulted in intermediate habitat requirements for unisexuals relative to that of their hosts. So historically, PLEE Island existed as several limestone outcrops, but the island has been drained with channels beginning in the late 1800s. In addition to wetland drainage, other habitat has been lost due to logging and land conversion for agriculture, quarrying, and development. Today, agricultural land is the primary land cover type, as shown in the map on the right in yellow, with forests and wetlands shown in green, restricted to small areas across the island, mostly in protected areas outlined in black. So our habitat suitability models revealed that suitability varied across the salamander genotypes. Unisexual diploids had the most available habitat in the map on the left, while unisexual triploids, those that have an extra texanum chromosome, had a narrower habitat niche, and the smallmouth salamanders had the least amount of available habitat, being mostly restricted to 3 locations across the island and being most associated with breeding sites and other wetlands. Generally speaking, using data from all of the salamanders, most of the island is unsuitable habitat, with suitable habitat being restricted to around 2.5% of the island. Suitability was primarily determined by the distance from a breeding site and being close to forested areas and wetlands, although these land cover types were considered to be unsuitable when they were far from a breeding site. Habitat suitability was found to markedly decrease immediately after leaving the breeding site. So these results, in addition to some connectivity work that we've also done, emphasize the need for improved connectivity between populations to ensure their long-term viability. Increased habitat protection and enhancement would also assist this unique salamander complex. So we were able to create some specific management recommendations to be sent to the stakeholders involved, namely the Nature Conservancy of Canada and the Ministry of the Environment. The Nature Conservancy of Canada has used these recommendations with the environmental conservation work pertaining to salamanders that they're currently doing on PLEE Island. They dug 3 new ponds in 2020 close to existing ponds to create a large wetland area, and 2 other ponds are being dug this year. All of these ponds are being built close to existing forests and will have saplings planted around them to help the development of a canopy. So you might have seen this pond previously in the presentations by Jordan and Evan. And NCC are also working on some deciduous forest regrowth in areas that were historically farmland and are close to existing salamander habitat. Most of these ponds, most of these ponds are in areas that are currently unsuitable, but would help to increase suitable habitat and connectivity between the populations. So overall, our findings emphasize the need for continued conservation efforts for these salamanders on PLEE Island to ensure the long-term viability of this salamander complex. And I think I'm running out of time, so I'll just quickly say that I'd like to acknowledge the people and organizations that made this project possible, namely funding through the Species at Risk Stewardship Program.
That connectivity model was quite fat with like how red it was. I mean, the suitability model.
Yeah. Yeah, connectivity was also pretty low, like suitability was. And basically all of the connectivity pathways between the populations were longer than could be realistically used by the ambystoma salamanders. So basically we just need to focus on conserving the areas where they are now and the few areas where connectivity could be improved.
Oh, here we go. Pam asked, what was the temperature difference in the constructed ponds versus the natural ones?
So for the one constructed pond that we look at, I don't know the exact temperature difference. I haven't quite gone through the data yet. We used eye buttons to measure both air temperature and water temperature across all of the sites. So at the one focal pond that was shown in the photos, it on average was a lot warmer than the others, mainly because it was out in the sun. It had forest cover on one side of the pond, but the rest of it was out in an old farm field that NCC is working to restore to a more natural, like wetland type site. So I don't know the exact temperature difference, but I expect it to be a significant difference because the larvae there were a very large compared to all the other sites. They were really small with the natural sites.